If you don't mind, let me share the screen first. Can you hear me now?
Yes.
Okay, good afternoon, everyone. It's time to start our class. My name is Kai. I come from the Gordon Center for Molecular Imaging of MGH. It's my great pleasure to have this opportunity to talk about the structure inherent targeting strategy in near-infrared fluor for the molecular design and development. So first, welcome to all of you to today's talk. And today we will focus on the design and then the application of the targeted optic contrast agent, ranging from the small molecule to nanoparticles for nanomedicine and imaging-guided surgery or disease treatment. So in the small molecule section, I will talk about the design of near-infrared fluorophores by using the strategy of structure inherent targeting. Today, the learning objectives include to understand the general concept of drug design and to understand the basic concept of structure inherent targeting and the physical-chemical property of the contrast agent, and also understand the targeting properties of the contrast agent. Today, my talk include this four part. Let's start from the concept of drug. When people talk about the drug, here the drug name is chemical drugs. So what is a drug? We know, I believe everyone takes the drugs or has the experience. So normally when people talk about a drug, it means the chemicals for the treatment or prevent of disease. Actually, the chemicals used for the diagnosis aims also belongs to the concept of drug. For example, the contrast agent in the clinics for CT, for MRI, for PET, and SPECT imaging for this medical imaging. The agent also belongs to drug. Among them, some of the near-infrared fluorophore had been approved by the FDA for widely used in the clinic. There are several basic but very important concepts in drug design. The first one is structure-activity relationship, which means the relationship between the chemical or 3D structure of a molecule and its biological activity. It is easy to understand that the structure of the chemical is the base of everything, not only the activity, but also the side or adverse effect and toxicity. Then what is a pharmacophore? This is another concept we will talk about many times today. It is one of several parts of the structure and necessary to ensure the activity of the molecule. Actually, pharmacophore of the whole drug molecule is a rational ensemble of steric or electronic features that are necessary to a specific biological target. The key of fragment-based drug discovery is based on the find and optimization of pharmacophore. Last concept is the Lipinski rule. You may have heard about the Lipinski rule or Lipinski 5 rule. Actually, it is a summary of the orally active drugs. It describes the number of hydrogen bond donors and acceptors, the molecular weight range, and then the log P values of these drugs. Of course, there are many exceptions of the drugs in the clinical use, but Lipinski rule is still very useful to remind us during the drug design. In the field of optic imaging, if you want your molecule to be more like a drug or drug candidate, or you plan to conjugate the dye with something, you need to be familiar with this the following concept. So the Stokes shift, it means the difference between the peak of absorption and emission. Quantum yield generally describes how efficiently fluorophore convert the excitation light into fluorescence. And the fluorescence quenching fluorescence lifetime means a measure of the fluorophore spent in the excited state before returning to the ground state by emission of the photon. These concepts are very important. So if you are interested to learn more details, please search the concept of these. During the past 20 or 30 years, actually a great achievement has been made in the field of optical imaging. By using the near-infrared fluorescence, we could see different organs into the brain, lymph node, thyroid, pancreas, kidney, ovary, bone, cartilage, nerve, and so on. From there, we are aiming to see people want to see the disease in a molecular level. For example, we could diagnose the cancer, different cancers in the molecular level. This is a summary, and a figure list of the most popular fluorophores from the UV range to the near-infrared range. From this chemical list, we could see a lot of chemical structures which are very popular. For example, the indocyanine-based structure, the BODP-based structure, the rhodamine-based structure, and the oxygen-based one. So they are all widely used in the lab to imaging, for the imaging purpose. So when you read the papers, there are some other concepts people frequently talk about. And people normally people always categorize the dyes under different name. If we categorize the dyes by different chemical skeleton, we could see, as I showed in the previous slide, we could see the rhodamine, BODP, cyanin by the chemical name. If we different them by the wavelengths of emission or absorption, we could see them in the UV range, visible range, or near infrared 1 or 2, these different windows. If people talk about the mechanism of the fluorescence, why this fluorophore caused generated the fluorescence, you will have more chance to see PET, HST, FRET, or AIE, this kind of word. So again, if you are interested, please check and learn these names after the class. And for the application, most of the fluorophore are special designed or conjugate with some targeting ligand for the purpose of special target. And also the application include the pH sensor. All these dyes are used for the detection of metal ions, for the hypoxia voltage. All these applications are widely used for this dye. Overall, the design consideration of optic contrast agent are very similar to the drug design. You need to think about this stability, the sensitivity, specificity, and safety of your molecule. But at the same time, when we designed the fluorophore, the near-infrared fluorophore, we also need to think about the optic property of the molecule, because the chemical and physical stability will cause the change of the optical property of this compound. So in this paper, this is a very good summary of the consideration when we design the optic contrast agent. So far, FDA has approved only 2 near-infrared fluorophores. One is named methylene blue, and another one is cyanin green. Methylene blue is a 700 emission fluorophore. ICG is 800 nanometers emission fluorophore. Although we only have these 2 in the clinic, but still there are many issues when using these 2 dyes in the clinic. For example, the most important issue of these dyes is the high background uptake. This chemical is named the ZW800S1. This is a very interesting structure. If you look at the structure, you will see the 3 negative charge maltes, and then the 3 positive quantum maltes. So this overall, this is a zetranic molecule. This is a new near-infrared fluorophore developed by our lab, and it is currently in phase 2 clinical trial. This is a non-targeted dye. When we inject this in vivo, there is no special uptake in vivo. It was designed, the COOH, the carboxylic group here, was designed for the conjugation with targeting ligand for special targeting. At the same time, these 2 sulfonate group here, they were designed and introduced to this skeleton to improve the water solubility of this molecule. If we inject this molecule into mouse or rat, it is, we could see it majorly metabolized through the kidneys with very low background residue in other organs. But for ICG and other dyes, for example, the CW800, it has 4 negative charge, but with 4 sulfonate groups here. The solubility is not a concern for this molecule, but the issue is it has a very high uptake in the after injection in vivo. We could see, except the kidney, the other organs include liver, intestine. It has a very high, relatively high background residue. So next, we are going to talk about the design of the contrast agent. So generally, most people use the traditional design for the development of contrast agent. So the left side, it needs a targeting ligand, which normally include the small molecules, including peptide, proteins, or polymers from the small molecule to big one, to drive the whole molecule to the target. There is normally an isolating linker between the targeting ligand and then the effector domain. Effector domain here means normally we use the fluorophore or any other radioactive to show the imaging of the whole molecule. Of course, to balance normally to adjust the BioV and the targeting ability of the whole molecule, we do some modification on the whole molecule to modify the size, the charge, and then the hydrophobicity of the molecule for better BioV and pharmacokinetics. This is a good example to understand the traditional design strategy. PSMA is a membrane protein, and it is expressed more in prostate cancer. QE here, this is the peptide targeting ligand for PSMA. When we conjugate this QE with different dyes we picked here, through a spacer. Here we choose the PEG linker. It is very common to be used as a linker between the targeting ligand and then the dye. So we choose the different dye here, and then we made the targeting contrast agent. The in vitro and in vivo data are quite interesting. For the in vitro data, we found that the charge and then the linker's length are very important for the PSMA binding. The negative charge from 4 to 6 are necessary to give a better imaging than the other charge numbers. And then also the linker's length is also important. We found the PEG4, the PEG linker with 4 repeated unit, with the PEG4 linker. It shows a better targeting ability than PEG2 and then PEG12, the other lines. On the tumor mice, this is the PSMA on the left side. This is the PSMA positive side of the tumor. On these mice, we could see the clear tumor targeting by using the QE PEG4 ZW800 plus 3C, which means the success of the molecular design. However, we should know that the traditional design strategy is popular, but it has its own limitation. Once conjugation with the targeting ligand and then the dye, the targeting ability and then the BioD character may still be governed by the targeting ligand, but sometimes may not. And we should treat the new compound as an entirety to investigate the bioactivity and then the targeting ability. Here is another example. The structure here in this green box, this is a molecule, it's called a PAM. This is a small diphosphate. It has 2 phosphate group here. This drug is designed for bone disease. After conjugation of PAM with different dye structure, these dye are different in net charge number and log D value. They are different. So we could see the background signals are different of these 3 molecules. We found the ZW800-1, this conjugation give a better targeting result and a less background residue compared with the other 2. From the structural activity relationship, we know the quantum amine here normally drive the molecule to the cartilage. And actually, we see this pattern. That means, except the PAM here, the 2 phosphate group direct the molecule to the bone. This part, the amine also played a role to bring the molecule to the cartilage. The third example is the CRGD conjugated with Psi5.5 and ZW801, our molecule. CRGD is a cyclic peptide ligand, and it is targeted for the alpha V beta 3 integrin, which is highly expressed on tumors. Using the similar strategy, conjugation with Psi5.5 and ZW800 could give the in vivo tumor targeting respectively. But similarly, we see ZW801, this conjugation give less background uptake compared with Psi5.5. Conjugation of dye with protein is very common if you want to see the in vivo BioD of the big molecule. Herceptin, this is an antibody drug for cancer, special for the breast cancer. When conjugation with ZW800, and then engineered by the PEG linker, it forms a micellar nano complex. The in vivo data also showed the good tumor binding and anti-tumor effect of this complex. Next, we are going to talk about what is structure-enhanced targeting. SIT means a single compacted flow for targeting, imaging, and therapy. The idea of structure-enhanced targeting is based on a series of discovery in our lab, and the concept of pharmacophore and fragment-based drug discovery. We could think about it in this way. If our goal is to see the target, we may only need the pharmacophore or the key chemical moiety to be conjugated on the skeleton of the dye, even without use of the linker. So with this basic, very simple hypothesis, we proved, we made several of the example molecules and to verify the concept works or not. So if this is in the green area, this is a skeleton of the inosine dye. We could see here if you have the learn the chemistry, organic chemistry, you will see like on this skeleton structure there are many positions for chemical modification and substitution. We could put the targeting ligand around and then at the same time modify the structure to control the BioD of the molecule. And we believe this kind of design will finally improve the drug likeness of the contrast agent. So this is the concept of structure inherent targeting. Again, let's go back to the bone targeting topic. Previously, we showed the bone targeting dye using the PAM conjugation by using the traditional design strategy. But if we look at the mechanism in a molecular level, we will know like this is the PAM the analog, but this is an example. We will know from the mechanism. We will know it is a OH group on the phosphonate group part, which are chelated with the bone matrix. This is the key for the bone targeting. If after we know this, we are thinking, why don't we just borrow this phosphonate group and emerge with our dye skeleton? So with this concept, we developed a couple of different dyes ended with the phosphate group. Look at the structure here, you will see the difference. It compared with previously molecular design, we didn't use any quantum amine here. This is to avoid the cartilage targeting. So by using this kind of design strategy, we could see the bone clearly the bone targeting signal in mice, especially by using the P700 and P800, the SOC, these two molecules. We get the similar result in the nude mice and then in pig. And one thing worth to note is that this bone signal is very stable. There we could see the signal still there after a month. Similar, let's go back to the PSMA project. This is a project we are still working on. We introduced the PSMA targeting molecule previously by using the structure inherent targeting strategy. We connect the KOE part to the dye directly without using the linker. This is the dimer form and this is the trimer form. Even without the linker, we could clearly see the PSMA targeting in the cellular-based assay. And this is very important because compared with our knowledge of the PSMA small molecule ligand library, this is a totally new structure-activity relationship. Next example, this is related with thyroid and parathyroid imaging. We know even for human, the thyroid and parathyroid are very small in our body, and they are difficult to be removed in the thyroid economy of the patient with a tumor in this area. We designed T700 and 800, these two molecules, to separately target to the thyroid and the parathyroid. The detailed mechanism is still unclear, but we believe the two phosphate fluoride groups on these two sides play an important role for the targeting. Next example is about the nerve targeting. During the surgery, we know nerves are very difficult to be recognized and avoid if you don't want to hurt the nerve during the surgery. So if we have a dye to be special for a nerve, it will be very helpful in the surgery. Oxygen, this is an important skeleton for near-infrared dyes. In our study, when we modified the chemical groups on this nitrogen atoms, oxygen 4, this molecule showed very good nerve targeting, and during the peak thyroid resection, the recurrent laryngeal nerve in peak was well kept after the resection of the thyroid. This is a good example for the SIT strategy. And I believe you guys already see this picture previously. This is a small molecule we find it is especially for the targeting of pancreatic neuroendocrine tumor. It is very sensitive for the detection, even for the ultra small ectopic tumors. We could see the tumors even less than 0.5 millimeters. And it is very fast. After injection in vivo, for a few seconds, we could clearly see the tumor light up. Similarly, what we could do based on that structure, by using the same skeleton, if you compare it with the structure with the molecule of the pancreatic tumor targeting, we used the same skeleton of the structure, but this time we want to do the dual model imaging. So we basically we want to introduce F18 into the structure and see the PET imaging for the pancreatic tumors. So by the video chemistry work, we could introduce F18 into the skeleton of the molecule, and actually we could see the tumor through the PET imaging. This is an example of the SAT design strategy. Actually, the dual model imaging, for example, the near-infrared PET, this is one of the future directions of medical imaging. People, more and more people are applying the dual model imaging for better targeting of the disease. Compared with other imaging modalities, near-infrared imaging is non-radioactive. It has high spatial resolution, fast and real-time display, and a relatively low cost and portability, except for multimodal imaging. near-infrared tool window. In recent years, people divided the near-infrared fluorescence into further two windows from the near-infrared one, which means normally covered 600 to 900 nanometers to over 1,000. This area is called near-infrared tool window, which means deeper penetration. This is another hot field in the future, and our lab is also focused in this area. In summary, no matter which kind of design strategy, optimized targeting is always a key, and there will be a huge clinic need for the targeted fluorescent contrast agent. And this is our mission to push our technique to be useful in the clinic. So that's all of my talk today. And in the end, I left 2 questions for everyone. Let's go through these questions all together. So the first one is, when design an optic contrast agent, what is the 4S criteria? It is easy if you remember the slide I showed. It covered the sensitivity, stability, specificity, and safety. It is very similar to the drug design, the consideration of drug design. For the clinical use, a contrast agent needs to be designed by considering the physical, chemical, and optical stability, and also the reasonable blood half-life, selective binding to the desired epitopes, and a low non-specific uptake in normal tissue. All these things we need to think about. The second question is, when design the structure inherent targeting infrared fluorophore, which volume parameters need to actually all of these items we need to charge, the polar surface area, and then even the root. Actually, all these concepts belongs to the Lipinski 5 rule, if you remember. Design of an optimal contrast agent targeted to a specific disease is needed for efficient imaging, diagnosis, and therapy. All these concepts, they are super important. And plus, the excitation and emission maximum, the quantum yield, the 3D structure of the contrast agent, and then the connection way of our targeting ligand, and then the dye skeleton, they all need to be considered when designing this molecule. This is all of my talk today. If you have any questions, please feel free to ask. Or you could email me after the talk if you still want to learn more details of the optical imaging. Thank you.
Thank you very much for your talk today. And yeah, if anybody has any follow-up questions after this percolates in your brain for a bit, please feel free to contact us. And so we'll take just a second here to switch over to our second lecture of the day. Let's see. Yeah, hold on. You have co-host, so you should be able to share.
Okay.
Yep. Yeah, we see your screen. Perfect.
Yeah, let me start. Okay, hello everyone. I'm Bao Kang, instructor at Golden Center of MGH. Today I'm going to talk about nanomedicine from small molecules to nanoparticles, especially polymeric nanoparticle based cancer targeting and therapy to cure the GIST tumor with image-guided surgery and drug delivery. Here's your learning objectives. Through this lecture, we will understand the general concept of nanomedicine and discuss what are difficulties, including biological barriers and trade-off in cancer nanomedicine, and discuss the physical chemical properties of the nanoparticles, and talk about the examples of the theranostic and clearable nanoparticles. And this is outline of my talk today. First of all, I'd like to a little bit talk about the background of the nanotechnology. In the 1959, Richard Feynman, who was a Nobel Prize winning physicist and the father of nanotechnology, gave a great lecture entitled "There's Plenty of Room at the Bottom." So in that lecture, he predicted that the nanoparticle will able to help and solve the problems in the chemistry and biology. So what are the reason of nanotechnology can do, and why the nanotechnology is important in the field of especially nanomedicine? So here is a scale for biomolecules compared to artificial nanoparticles. So as you can see here, the size of the nanoparticles are pretty similar with biomolecules such as virus, proteins, and DNAs. So if we can manipulate such nanoparticles, we can specifically identify, target, and treat the disease. And nanoparticles have unique physical chemical properties. For example, it has high surface to bulk ratio, which means surface property is more dominant in entire properties than that of the bulk state, and it can very active. So we can use it as a catalyst, and we could get high sensitivity in diagnostic applications. However, the nanoparticle can easily make aggregates, and it can be toxic because of high reactivity if they are unstable in the body. And also, nanoparticles have unique optical properties depending on their size. So using these unique properties of nanoparticle can be applied to in vitro diagnosis as well as in vivo bioimaging and therapeutics. For in vivo bio applications, many researchers focus on the cancer diagnosis and treatment because of EPR effect, enhanced permeability and retention effect. So target mechanism of cancer is based on the EPR effect. So as you can see here, the healthy blood vessels have narrow endothelium junction. However, the tumor angiogenic vessels show leaky vascular structure with large intracellular pores. So when the nanoparticles with optimal size are injected intravenously, nanoparticle can penetrate much more into tumor tissue through the leaky vasculature than in healthy tissue. And then nanoparticle can retain in the tumor for longer time because tumor has lack of function, lack of functional lymphatic vessels. In the case of the active targeting, ligands have affinity to specific receptor used like this. So even though we use targeting mechanism, passive and active targeting strategy, specific cancer targeting is not very successful because there are several biological barriers such as reticular endothelial system and endothelial and epithelial membranes, complex networks of the blood vessels, and so on. So to overcome biological barriers, when the nanoparticles are used in the body, we need to consider the size, shape, and surface property of the nanoparticle, which greatly affect to the clearance and biodistribution and tumor targetability. So as you can see here, the biodistribution in the major organs varies depending on the physical chemical properties of the nanoparticle, like size, shape, and charges. So this video more clearly shows the effect of the physical chemical properties of the nanoparticle. There are two nanoparticles, the quantum dot with different sizes. One is 27 nanometer and the other one is 5 nanometer in diameter. So bigger size of the quantum dot is excreted through the hepatobiliary route. However, the small size of the quantum dot was able to clear through the urine like this. This is another example, especially single wall carbon nanotubes. CNT mostly accumulate and stuck in the liver, so it can induce the potential toxicity. So this kind of non-specific accumulation and off-target drug toxicity are the main problems of the nanomedicine. So now I'm going to talk about optical imaging. As you know, but there are many imaging tools such as PET, SPEC, CT, MR. Among these optical imaging, imaging tools, the optical imaging is one of the important tools for real-time imaging. And optical imaging has several advantages, such as high spatial resolution and sensitivity and high multiplex capacity and so on. So, but the major challenge in the in vivo optical imaging is the limitation of penetration depth because of tissue absorption and scattering. However, if we use longer wavelengths from 650 nanometer to 1700 nanometer, near infrared region, we can avoid the tissue absorption and scattering and have a better resolution like this. So if we use optimal imaging system, And contrast agent, we can visualize the lymph nodes, the mesenchymal lymph nodes, and cancers. In addition, we can investigate the tumor microenvironment using a microscope system like this. So, okay, so let's go back to the nanoparticles. Now the non-specific accumulation and off-target drug toxicity are the main problems. So what is the ideal case? For biodistribution, the nanoparticle should go to the target, and then the rest of the untargeted nanoparticle should be excreted through either hepatobiliary or urinary excretions. However, we believe renal clearance is more favorable because it's a faster clearance rate. For ideal pharmacokinetic properties, nanoparticles should distribute rapidly into the central compartment at the initial state, and then they should have high enough concentration in the bloodstream so it can have a large area under the curve, which means it has a higher chance to meet the target. After a certain time point, it should be cleared out completely like this from the blood. So to investigate this, first we systematically varied the size of polymer, polyethylene glycol polymer, PEG, from 1 kilodalton to 60 kilodalton, and investigated the size dependency in terms of biodistribution, clearance, pharmacokinetics, and tumor targetability. So as you can see here, the larger nanoparticles stay longer in the bloodstream, and accumulate in major organs because they circulate for longer times. Next, we tested tumor targetability. The signal was significantly increased in the tumor site as polyethylene glycol size increased, but the background signal is still remained after 24 hours in the larger size of polyethylene glycol nanoparticle group. If we do the therapy, larger size of nanoparticle could cause potential toxicity. So this result clearly shows that the 20 kilodalton polyethylene glycol is balanced in between tumor targetability and background signal, and it has lower potential toxicity. Based on the previous result, we designed a theranostic nanoparticle for image-guided surgery and drug delivery. This is the structure of balanced charged nanoparticle, which has digital ionic surface. So we start with the small size polymer polyethylene lysine, which is 4 kilodalton. We attach the cyclodextrin for drug delivery and add the NIR44 as an imaging domain. So the final hydrodynamic diameter is still within the renal filtration threshold, 5.5 nanometer. So it is renal clearable and it can avoid non-specific organ uptake, resulting in lower toxicity. We named this nanoparticle H dot, which stands for Hubble dot. So this is overall schematic synthetic scheme. So I'm going to skip the details. At the end, we change the surface charge of the H dot to positive, negative, Jupiter ionic, and no charge to confirm the charge effect. So here is the biodistribution data. So Jupiter ionic H dot shows very low liver uptake compared to positively charged H dot. And also in the pharmacokinetic study, Jupiter ionic H dot shows the best performance for clearance and potential tumor targetability. For the proof of concept, we chose GIST, gastrointestinal stromal tumor, as a target disease, which is the most common mesenchymal tumor in the GI tract, and imatinib is used as an anti-cancer drug for treatment of GIST. Imatinib can make an inclusion complex with the cyclodextrin on the H dot by hydrophobic interactions, and it can release under acidic environment similar to the tumor microenvironment. And this imatinib can bind to the KIT receptor on the GIST tumor cells and act as a tyrosine kinase inhibitor, which can induce the apoptosis. So after loading the imatinib onto the H dot, we confirm the drug release under the acidic condition, like pH 5, and we confirm in vivo therapeutic efficacy of the imatinib loaded H dot. The therapeutic efficacy is similar with imatinib itself. And we investigated the biodistribution and tumor targetability of the H dot. The NIR dye conjugated imatinib and imatinib loaded H dot were injected into the tumor bearing mice, and imatinib itself didn't show high tumor uptake and accumulate in the liver and spleen due to the hydrophobicity of the imatinib itself. But in contrast, H dot can deliver the imatinib to the target tumor and didn't show any significant signal in the major organs except the kidneys. And we did the pharmacokinetic study, and in this result, volume of distribution of H dot is very similar with the volume of the extracellular fluids, which means it doesn't have any non-specific distribution. Then we did image-guided surgery using the H dot, and we can completely remove the tumor by image-guided surgery and then small residual tumors after the first resection by image-guided surgery, like this. And we also confirmed the therapeutic efficacy of the H-dot. The signal intensity is pretty much correlated with the injection of the H-dot, and tumor size was successfully suppressed with the imatinib H-dot treatment compared to the tumor size with the imatinib alone treatment group. In the H&E staining imaging, we were able to observe many empty space, which means imatinib can induce the apoptosis, and we confirmed the NIR signal using microscope imaging and found that H-dot can penetrate deep into the tissue because it's small size and non-sticky property. So this is another example of renal clearable nanoparticle. The renal clearable nanoparticle can be used for heavy metal detoxification. So in this case of the ion chelation therapy, by conjugating the small molecule ion chelator diferoxamine, DFO, onto the polylysine backbone, and then we can make a nanochelator. So once the nanochelator is injected, it can systematically circulate and can bind excess ion in the body, and then remove the ion by clearing out to the bladder, like this. So this is summary of my talk. So nanotechnology and nanomedicine open new concept of the cancer imaging and therapy, but we still need to understand the biological barriers and trade-off. And the enhanced permeability and retention effect is a crucial concept for the tumor targeting, but it's still controversial. And physical chemical properties determine the fate of the nanoparticle in the body, and the nanoparticle should have favorable clearance to reduce the background as well as potential toxicity. And we found the renal clearance is more preferred. And potential toxicity and unexpected adverse effect of the nanoparticle have to be solved before clinical translation. So this is all my talk, and I also have two simple questions for you. So this is first question. So nanoparticle composed of organic or inorganic materials size between 1 to 100 nanometers. So which of the following is not classified as a nanoparticles? A, albumin, B, dendrimers, C, fluorescein, D, fluorines, E, quantum dots. So correct answer is C. C is a small molecule, and this is actually fluorophore in the visible range. So this has a hydrodynamic diameter less than 1 nanometer because of its small molecule. The others, this is proteins, and this is polymer, branched polymer, and this is kind of carbon nanostructure and quantum dot semiconductor nano materials. And next question is, when our excretion is mostly limited by hydrodynamic diameter and surface charge and hydrophobicity of the injected substance, so which of the following is cleared by kidneys? So A is herceptin, antibody, and protein, serum, human serum albumin. C is ICG, Pro4, and D is aptamer, and E is red blood cell. So correct answer is D, aptamer. This is the size is around 3 to 5 nanometer in hydrodynamic diameter, and it can clear rapidly from the bloodstream, and the half-life is minutes to hours. And the others, this is serum protein. The size is around 11 nanometer, so it's bigger than the kidney threshold, and antibody as well. And red blood cell is 6 to 8 micrometer in diameter, so it cannot pass through the kidney filtration. Okay, this is all my talk. Thank you for your attention.
Well, thank you very much,